The hormone irisin is necessary for the cognitive benefits of exercise in healthy mice and can rescue cognitive decline associated with Alzheimer's disease, according to a study published August 20 in Nature Metabolism. According to the authors, these results support the hypothesis that irisin undergirds the cognitive benefits of exercise, a link that has been long debated. In addition, this study has paved the way for thinking whether irisin could be a therapeutic agent against Alzheimer's disease, says biologist Stefan Mock with the Leibniz Institute for Farm Animal Biology in Germany, who has been critical of the methods used to study irisin in the past and was. Many studies have found that exercise is good for the brain, but the molecular mechanisms responsible for the cognitive boost have remained elusive. During her postdoctoral studies, neuroscientist Christiane Rand found that the gene that codes for irisin becomes highly expressed in the brain during exercise, one of the first studies linking irisin with the brain. The result demonstrates that loss of memory is faster in this Alzheimer mouse model if the mice don't have irisin, Rand writes in an email. If a lack of irisin worsens memory loss in an AD model, could a boost of irisin improve cognitive function? The authors set out to answer this question with a gene therapy approach. Using a viral vector, the researchers boosted irisin production in the livers of their AD model mice, which translated into increased levels of the hormone in the mice's blood and also in their brains. As the viral vector only increased irisin mRNA in the liver, the authors concluded that peripherally produced irisin was able to cross the blood-brain barrier. The data presented in the paper convincingly show that peripherally administered irisin at pharmacological doses has really beneficial effects on cognition, Mock writes in an email to the scientist. He adds that the relevance of exercise in this context is not clear. Irisin may have therapeutic potential at high concentrations. I doubt whether pharmacological concentrations may be reached by exercise. How irisin, and especially peripherally delivered irisin, acts on the brain is not yet settled. These different pieces of evidence support that irisin has an effect directly on astrocytes and possibly microglia, says Rand. Meanwhile, in the irisin-deficient mice, the authors saw reduced dendritic spine density and smaller dendritic spine heads on adult-born hippocampal neurons, indicating that the neurons were developing abnormally. Whether the effect of irisin on GLIA influences adult hippocampal neurons is an interesting, exciting question which we are going to work on right now, she adds. I think this study opens this new field. Sergio Ferreira, who specializes in biochemistry, biophysics and neurobiology at the Institute of Medical Biochemistry at the Federal University of Rio de Janeiro and was not involved in this study, tells the scientist. It's very nice that the authors find that irisin appears to act by modulating or cooling down these glial cells and preventing the exacerbation of inflammatory responses, which may actually be damaging to the brain. Ferreira published a 2019 paper reporting that irisin is important for memory. The initial discovery of irisin, made in 2012 by a team led by Bruce Spiegelman, a co-author of the current study, showed that irisin is elevated upon exercising, and acts on white adipose tissue to convert it into calorie-burning brown fat. In the new study, there was no effect on adipose tissue of any kind, in sharp contrast to previous results, Mock says. Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.